Okay, hello. Okay. Uh, so right now, let us continue with the wait. Let me. Okay. So can you hear my voice? Can you see my screen?
Yeah, I can hear
Okay.
screen.
Yeah. So if you're okay, okay. Let us continue. Okay. So first one. Okay, we will. Last time we learned until how to draw the glucose, right? Alpha glucose and beta glucose. Okay. So right now, let us look at the continue part. Okay, the below part. Okay, so okay, we need to know polysaccharide. We got three types, right? Okay, we got starch, we got glycogen, okay, and we got cellulose. So for starch, starch is made up of alpha glucose. Okay, starch, they are actually made up of alpha glucose, and they does not contain any beta glucose. And glycogen also made up of alpha glucose. So what we can do a summary here is starch and glycogen, both of them are made up of alpha glucose, but for cellulose, they are made up of beta glucose. Okay, so this is the first one. And later I'll do a recording now, okay? Then I upload the recorded lessons to my YouTube, then I share you the YouTube link. Okay. Then and for this Wolf meeting, right? They got free 40 minutes lessons. So later when they jump out of the meeting after 40 minutes, you can re-enter the same link. Okay. Then but the recording they will continue. Okay. Yeah. So let us look at the first part now. Okay. So this is what we gonna learn today. Okay. So remember, starch and glycogen, they are made out of alpha, while cellulose is beta. So let us zoom in. Okay, so let us zoom in. Okay, biomolecule key terms, and okay, and the tips here you see, examiner tips and tricks. So you need to know how to recognize and draw alpha and beta glucose. So let us continue. Okay, biological molecule key terms. Okay, what are the key terms here? So the key terms here is, okay, you need to have lipid. Okay, we need to learn a few function, which is carbohydrate, protein, lipid, nucleic acid, and water. So I hope today we can settle these two lah. Okay, and then we will continue the other one. So now first one, okay, this one have a brief introduction about what you are learning lah. So first one, okay, you will have lipid. So lipid, we call it triglycerides. So when triglycerides break down, you will form glycerol and fatty acid. Okay, and nucleic acid, their monomer is formed by nucleotide. Okay, so carbohydrate, your monomer is glucose. Protein, your monomer is amino acid, while lipid, your monomer is glycerol and fatty acid, and nucleic acid, your monomer is nucleotide. So this one is the biological molecules diagram. Okay. Yeah, so let us continue with the below part. Okay, and you contain something called inorganic and organic. So how do you differentiate? So for organic, is all of them contain C and H. This one you will learn in chemistry. Okay, organic chemistry and inorganic chemistry. But biology, organic part means that you consist of C and H. You see, carbohydrate, protein, and you get polypeptide, amino acid, getting smaller and smaller unit. Carbohydrate, you start from polysaccharide, but the thing you will get is monosaccharide. That's why you get smaller and smaller. So anything contains C and H we call organic, and the remaining one does not contain carbon and hydrogen, we call it inorganic. So these are the biomolecules that found in your living organism lah. Okay, so this one I leave it to you because not hard. And the main thing is I go through this one with you. So for For the part that is organic, they contains carbon and hydrogen. And each carbon atom can form 4 covalent bonds. Okay, I'm not sure whether you learn chemical bonding in your chemistry already or not. Okay, but carbon, remember they can form 4 covalent bonds. So the example is what? Example, we call it methane. Methane gas is a type of alkane. The formula is CH4. So you see, one central carbon atom can bind to 1, 2, 3, 4 different elements, and we call it covalent bonds. Why? Because covalent bond is between non-metal binded to non-metal. Okay, so this is your non-metal. We form covalent bond, and normally carbon atoms, they will form a lot of different type structure. First one is we call straight chain. Second type is branch chain, while the third type is rings. You'll form a few different chains, different branch chains, or different rings. So this one is what we call carbon atom. And single subunit we call monomer. Okay, so if you have many repeating subunits, which we call monomers, so means that a lot of monomer join together, you will form polymer. Okay, you will form polymer, and the process of this we call it polymerization. Okay, the process here we call it polymerization. So this part, is it okay? Okay, yeah, so if this part okay, I'll continue below part. Okay, so let us look at carbohydrates. Okay, so first thing, carbohydrate, you contains CH and O. Okay, you contain CH and O. And the H and O atoms, they are always present in the ratio of 2/1. Okay, you contains in the ratio of 2/1. So 2/1 means that your H and O will have more number. Okay, so you see, I got H2O. Okay, I got H2O, and X and Y is the number of the each molecules present lah. Okay, so this is for carbohydrate. So the first class we will be learning is carbohydrate. And this one, I think majority of it you learn in IGCSE lah. So I'll just go through first. Okay, carbohydrate first type, you got monosaccharide. So monosaccharide is a single sugar. Last lesson we learned before, single sugar. And normally they are reducing sugar. While disaccharide is like you formed by 2 monosaccharides. And between glucose and glucose, we call it glycosidic bond. Okay. Example, this glucose and this glucose forms together. Okay, this one we call it disaccharide, since we only have 2 glucose molecules, while the middle part here will be our glycosidic bond. Okay, our middle part here will be glycosidic bond. While polysaccharide means that you form by many monosaccharides. So always remember, the process of forming a long chain, we call them condensation reaction. Condensation reaction. So condensation, you join two, means that you join two sugar or more by removing water molecules. Okay, by removing water molecules. And we got another process called hydrolysis. Okay, let us do some revision. So hydrolysis is you break two sugars by using water molecule. okay, by using water molecule. So this is monosaccharide, disaccharide, and polysaccharide. So the examples, let us go through. Monosaccharide, you got glucose, ribose, and glycerol aldehyde. Okay, and each contain their own specific number of carbon. While disaccharide, you will have maltose, sucrose, and lactose. Okay, maltose, sucrose, and lactose. So you see, the bracket is like they combine which glucose with which glucose in order to form this. And polysaccharide, you will have cellulose, starch, and glycogen. Okay, so each different types of classes, they got their own function. Okay, so first one, your monosaccharide, they are used in energy in respiration. Means that they provide energy for us, okay, in our daily life. Because they are the simplest sugar and they can act very easily. Secondly, we got building blocks of our polymers. So monosaccharide, they are actually forming a lot of polymer. Like example, your starch, okay, or your glycogen, they are formed by alpha glucose, right? Okay, then your polysaccharide, they are formed by beta glucose. Okay, so this is the table here for carbohydrate. First one, I think, yeah, first one, because this chapter is about carbohydrate and also our protein actually. Just that they play with different, they're using different notes.
Okay.
Yeah. Then after that, if you're okay, I think we need to fast a little bit lah. Okay. So yeah, so here we call it covalent bond. Okay, so this part I'll just go through because this is more on chemistry. You will learn more on chemistry about covalent bond. So covalent bond, okay, they are actually formed by what? You will use a word called sharing of two or more electrons. And I do a revision with you. Ionic bond is what? They will use transfer of electron. Okay, they will use something called transfer of electron. So electrons, they can be shared equally, forming a non-polar covalent bond. Okay, non-polar covalent bond to form a polar covalent bond. Okay, so what is non-polar and polar? So you can remember non-polar is they got the same electronegativity. Okay, or you can say non-polar to be simple lah. It's like a neutral molecule. Polar means that they can form charge. Okay, they can form charge. Yeah, so this one is in your chemistry lah. Okay, so I leave it to your chemistry teacher or anything. I just give you a brief, simple explanation or overview lah. Okay. Yeah, so covalent bond, they are actually very stable. Okay, they are actually very stable. Okay, and they need a lot of energy to break the bond. So sometimes covalent bond, they can form double bond. Okay, especially in unsaturated fats, or they can even form triple bond. Okay, so this is the covalent bonding. Okay, yeah, it's like a sharing lah. Okay, so this one you just read through. Okay, because you will learn more in chemistry. Okay, and then now we move on to condensation. So condensation, okay, is like dehydration synthesis. Okay, why? Because you will lose water. Means that if you want to form two things together in your condensation, you need to throw out the water. Okay, you need to throw out the water. So when your monomers combine together by your covalent bond, they will form polymers. Okay, so meanwhile your water is removed. Okay, so with these words is not enough to show you. Okay, let us look at this picture, then you will understand the whole thing. So you see, I got three monomers here. So if I want to form a bond between these two monomers, so I put monomer 1, 2, and 3. So example, if I want to form 1 and 2, okay, I need to remove one water molecules. And then between 2 and 3, I need to throw another 1 molecules. So if you observe, you will form 2 water molecules here between 3 monomers. So the formula is like that. If you got 100 monomers, so the number of water molecule, you minus 1. Okay, so you will form 99. If you got 2 monomers, so you will form 1 water molecule. Or if today you got 150 monomers, you will form 149 water molecules. So this is the concept here. And you see, once they form, okay, you see, H, this monomer, the one monomer throw H. Okay, one monomer, they will throw the H. Another monomer, they will throw OH. So H and OH will form one water, and they will be combined by glycosidic bond. Please remember this one. For carbohydrate, we have glycosidic bond. Okay, so this is for condensation. Yeah, this is for condensation. Condensation means you throw water and you combine them together. So, okay, hydrolysis. Yeah, hydrolysis is what? Okay, you got this equation, right? Okay, from left to right, we call it condensation. So, from right, you go back to left, we call it hydrolysis. It's like a inverted concept. So hydrolysis, you need water. Okay, lys mean break, hydro means water. So your covalent bond are broken when water is added. So you see, I add water to this polymer, then I separate them. So it's the same thing. If you use 2 water, right, means that how many monomers you will get? You will get 3 monomers. Okay, because just now we mentioned, if I got 100 monomers, I combine, I get 99. So if I use 99 to break this polymer, of course, I will get back 100 monomers. So this is condensation and hydrolysis. Okay, so for this part,
Okay.
is it okay?
Yeah.
Yeah, okay now, okay. Yeah, I think here still okay. So I'll just continue now, okay? And then, yeah. Okay, this is the summary of the bond. Okay, please take note, because they ask in MCQ. Okay, they like to ask in MCQ. So carbohydrate, you will use glycosidic bond. Protein, you will get peptide bond. Okay, peptide bond. So this is, okay, in chemistry, organic chemistry, you will learn C double bond O and H, we call it amide. Okay, please write down. This one you will learn. And amide is a basic group. Means that they are not acidic. They are slightly a little bit alkaline. Okay. And lipid, you will use something called ester bond. So ester bond is COO. Okay, COO. And you got 2 R group lah. We form ester. And how you form ester in chemistry? Carboxylic acid added with alcohol. You will form ester. And nucleic acid, between what? Between your nucleotide and another nucleotide, they are actually joined by phosphodiester bond. So this one, please remember. Okay, you put 1 star. Okay, you put 1 star here. Because MCQ, they would like to ask this one, and many of them confuse. Or sometimes they will ask for structure, 1 mark. They say state the bond present in carbohydrate. So you need to write glycosidic bond.
Okay.
Yeah. Okay, then after that, okay, let us look at this one. Okay, reducing sugar and non-reducing sugar. Yeah, this one I think last lesson we mentioned also. So only sucrose is non-reducing. Okay, the other one is reducing. So I think this one I let you read through, and they are actually related to food tests. Yeah, so please do some revision. Okay. Then yeah, I think it's not a problem for you lah. Okay, so I just leave it here and I'll skip to another one first. Yeah, and we also know how to form glycosidic bond. Yeah, so glycosidic bond, I directly discuss this diagram. So okay, you are given to, okay, this is what? This is called alpha glucose. Why I know? Because my OH is below. Okay, my OH is below. Always remember when OH is below, we call it alpha. And when OH is at upper part, okay, is at upper part, we call it beta. Okay, we recognize this part. Okay, so you see between two alpha and alpha glucose, how I gonna join them together? Simple concept. One glucose throw OH, another glucose throw their H. Okay, no matter which one, okay, you just throw either one will be okay lah. Here throw H, here throw OH. So this blue color H2O, you see, I managed to form water. Okay, I managed to form water and I combine them together. So you see, you will get one O, oxygen remaining here. And these two of alpha glucose, they are being combined. So monosaccharide, monosaccharide, I form disaccharide, which is maltose with water. And this bond, we got one name. So what is the name? Okay, it's like your chemistry, how you name in organic chemistry. Okay, so alpha, number in front, 1, 4. Why? Because they are connected to the first carbon of your alpha glucose and the fourth carbon of your second alpha glucose. So you see, 1 is here, here is 4. So we call it 1, 4 alpha glucose. Or we call it glycosidic bond. Okay, this one should be correct because we are mentioning the bond here. Okay, so if you have more, you will form a lot lah. You will form a lot behind. It's like a straight chain. Okay, they are actually like our straight chain.
Okay.
Okay, yeah. So this is our glycosidic bond, and then okay, sometimes they are actually catalyzed by enzymes. Okay, what is catalyzed? Catalyze means speed up. Speed up by catalyst. And one of the catalysts in biology, we call it enzymes. Okay, you will learn in another chapter. Okay, this one is like roughly chapter 3 if not mistaken. Okay, and then yeah, same thing, glycosidic bond, OH, OH, you throw one H from OH, so you join together. So if you are, okay, you see, if you are joining alpha glucose and beta fructose, you'll form something called sucrose. Okay, so fructose, if you got time, you can remember roughly remember the structure, how to draw. But alpha and beta glucose, you must know. These two must know. Other one, you can just read through. Okay, other one you can just read through. So this is actually our glycosidic bond formation. So normally you need to know how to draw alpha and beta lah. This is the most basic one they will be asking in A levels is alpha and beta glucose. Okay.
Okay.
And then after that, okay. Okay, there is a new thing here. Okay, there is a new thing here. We call it amylopectin. Okay, this one a bit related to your starch. Okay, so this one I'll use some time to explain to you. So glycosidic bond formation diagram. Okay, we will have something called amylopectin. So amylopectin, they are actually formed by two straight chain, 1 and 2. So the first straight chain, okay, amylopectin very special is what? Okay, you see here I got two chains, right? 1 and 2 here. Okay, 1 and 2 here. Okay, so we focus, okay, just now we learned what? We learned 1 and 4, right? 1 4 1 4 1 4 1 4 1 4 1 4. But right now they got a special thing here. Between your chain and chain, middle here we got one called 1,6 glycosidic bond. So you can imagine this is your straight change. Okay, you can imagine a stem lah, okay, a stem of the plant, and you suddenly got one branch out. So the branch, you will use 1,6 glycosidic bond. So this one, is it okay?
Yeah.
Yeah, okay. Okay, so if you're okay, and this is the summary. Okay, summary of the glycosidic bond inside your sugar. So we got maltose, sucrose, and we got cellulose, amylose, amylopectin. So this is your sugar. Okay, so you can identify, make sure you know how to identify where your glycosidic bond is in a carbohydrate. Okay,
So I will have to memorize all of this, right?
yeah, you need to memorize this one.
Okay.
Yeah, if you know this one, sometimes you answer the question, you no need to proof check so much lah. You can directly know which answer is correct.
Oh,
Yeah.
okay.
Yeah. Yeah, that's why I say this save my exams notes is quite good lah, I think. Instead of reading the textbook, right? Yeah, I got the textbook also, the green cover page one. That one I don't like. Too much word that one. Yeah, that's why I use this one lah.
Okay.
And normally they got write examiner tips and tricks, right? So when you revise the notes, you can take note this part. Means that the things that you need to know before your exam, or during your revision. Okay.
Okay.
Yeah. Then I think, okay, breaking the glycosidic bond is the same lah. You just add water. You add water and they get back their OH and OH. So it's like okay, you can remember, you've 1 plus 2, you get 3. This is condensation along with water. So when you use water to break this thing, you can get back the remaining 2. Yeah, this one is the summarize of it lah. Okay, although here a bit detailed, the summary you can do is when you add water, so you can break this bond and they will form back the initial things they have. Same goes to the below part. I add water into here, I get back what I want. Okay, so this is the first part of today's lesson. Okay, so far, am I teaching too fast?
No, I can catch up.
Yeah, okay. So, okay, your school teach until which part? Which chapter actually? I forgot to update from you.
To be honest, I am homeschooling right now.
Oh, homeschooling, okay.
Yeah.
So means that you are taking as private candidate lah.
Yeah.
Oh yeah, oh, okay. Then I think okay lah, I think okay. Because I'm not so stressed lah, at least because some of the uni they rush the syllabus is like, how to say without stopping one. Okay, that's why I scared I teach too slow or teach too fast lah. Okay, yeah, so okay. If here okay. Okay, I think we continue this page. Then after amylose this part, we take a short break, then only continue. So I continue the first part first. So we have learned to the final part lah, okay, which is a new thing in your A levels. You didn't learn in IGCSE one. Okay, you will learn something called amylose, and second thing is amylopectin. Okay, but no worries, this one I'll do a summary for you later. Okay, so first one, you got starch and glycogen. So starch and glycogen, okay, your structure and function you need to know. So both of them are polysaccharide, okay, and they can be branched or unbranched. They can be folded, or they can be straight or coiled. Okay, they are like actually, how to say, a lot of pattern, okay, a lot of structure, just like chemistry. Okay, you can be branched out, you can be folded, or you can be straight or coiled. So this is their starch and glycogen. So, okay, starch and glycogen, we call it storage polysaccharide because they are, okay, we call it storage polysaccharide. Means that they store something in our body, okay, due to two characteristics. First one is they are actually compact. Compact means that they are very, how to say, they are very high density. Okay, they are very high density, and they fit into a small space so that you can store large volume. Okay, you can store large volume. So that example lah, in Malaysia we eat a lot of rice, right? Okay, rice. So rice contain a lot of starch, right? So they will store as starch molecule, and they will slowly digest, slowly digest, slowly digest. So your starch will go from polysaccharide to disaccharide, okay, disaccharide to monosaccharide, and then mono, they will give us energy. That's why eating too much starch is not so good lah, okay? But for daily needs, you need some starch also, okay? You need some balanced diet also. That's why they are actually like our power bank. So example, if your phone don't have any, you are outside, okay? But your phone don't have any battery already. So the starch is like the power bank. They will give you some energy to charge your phone. So this is first thing, they are compact. Means that they can fit into a small space, and they can be in large volume. Like example, our power bank, their storage is like 10,000 10,000 mAh, okay, the unit, or 20,000. Yeah, so this is compact. Second one is insoluble. So they do not dissolve in cell cytoplasm, and they have no osmotic effect. Osmotic effect you will learn in a chapter called osmoregulation. Means that how your urine is produced and how your body maintain the water balance. Yeah. Because you got heard before, right? Hypertonic, hypotonic, isotonic. Yeah. This is what we call osmotic effect. Means that how we study how the water move from here to another place. This one is called osmotic effect. Okay.
Okay.
Yeah, and then let us look at the starch. Okay. Starch, okay, they act as storage polysaccharide of plants. So sometimes you eat, okay, sometimes you eat the corn starch, okay, or sometimes we will have like potato starch. That's why these are the starch inside our plant. Okay, and then they will store as granules. Okay, granules means large solid particles. Okay, granules is a larger form of particles, while smaller one you will have powder. Okay, you have powder. This one important also because in chemistry, especially rate of reaction, that chapter, you will encounter granules and powder. So they will ask you which one has a higher rate of reaction. So the higher rate of reaction is powder lah. Yeah. And due to many monomers, Okay, it will take longer to digest than glucose. That's why eating starch, you won't directly get energy. That's why sometimes after eating lunch, we will feel tired, right? Why? Because your body is digesting the starch so that you can get energy. So maybe after 30 minutes or 1 hour after your lunch, you will get energetic. Yeah, so this is the concept lah. And our starch is constitute and constructed from two different polysaccharide. One is called amylose. Okay, one is called amylose. Another one is called amylopectin. So this one after break we will enter this part. Okay. Yeah. So we take a short break until let me check. Now is 08. Okay. 3:14 we come back. And later after break, you can re-enter the meet. Okay. Yeah, so right now, let us continue. So we will be learning this one called amylose and amylopectin. Okay, so amylose, okay, they are actually formed by unbranched helix chain. Okay, they are formed by unbranched helix chain that consists of alpha glucose. Okay, that consists of alpha glucose, and they are stabilized by your hydrogen bonds within a molecule. So what is hydrogen bond? So hydrogen bond means that you is bonded with F, bonded with O, and bonded with N. We call it HF, HO, HN. Fluorine, oxygen, nitrogen. Okay? Yeah, so amylose, they are actually unbranched. Okay, 10, okay, you consist of 10 to 30% of your starch. Okay, you consist of 10 to 30% of your starch. So they are actually unbranched helix shape with 1,4-glycosidic bond. Okay, with 1,4-glycosidic bond between your alpha glucose and your beta glucose. Okay, between your alpha glucose and your beta glucose. So this can cause them to be more compact and more resistant to digestion. Okay, because you don't want to digest so fast, or else you will keep on eating. If you digest too fast the starch, maybe after lunch 30 minutes or 1 hour, you feel hungry again. Yeah, so we don't want this to happen now, okay? Or else we will overeating. Okay, so you will have this structure, which is you do 1,4 link alpha glucose molecule together by condensation. So they are actually a straight chain, and inside they consist of 1,4 glycosidic bonds. Okay, so this is your amylose. So you will, okay, in your starch lah, you will have less, okay, in starch, okay, in starch you will have less amylose, okay, and you will have more, okay, and you will have more amylopectin, okay, and you will have some low amylopectin lah, less lah, less amylose, more amylopectin. So this is amylose. So what is amylopectin? Amylopectin, they also, okay, they are actually 1,4 glycosidic bonds, but they will have 1,6 glycosidic bond and creating a branch molecule. So remember or not? I go back here. Yeah, this one, amylopectin. Remember or not? We just now got one straight chain, right? Okay, so you can imagine the yellow color one I highlight here is actually our amylose. Okay, this is actually our amylose. So amylose, if you got another branch and form another chain, we call it amylopectin. Okay, we call it amylopectin. So amylopectin, you will have one branch. Okay, you will have one branch. Okay, and you form, the branch is connected by 1,6 glycosidic bond. Okay, so amylopectin structure, you see, it looks like this. Just now I let you see that one. So you see they will have branch, and each of them they got terminal glucose molecule. So terminal glucose molecule, right, they are actually they are actually prevent them from forming forming glucose lah, to make the chain or the branch too long. Okay, so you have a terminal glucose, you terminate the production and fusion of your glucose molecules. Okay, so for this part, is it okay? Can you understand?
Yeah, it's okay.
Okay. Yeah, so I'll do a short summary lah, okay, between your amylose, okay, amylose along with your amylopectin. Okay, we will amylose and within your amylopectin. So amylose and amylopectin. So amylose, they are actually what? Okay, they made by condensation of 1,4-link alpha glucose. Okay, so amylopectin also the same. Okay, amylopectin also the same. While second one, we will have what? We will have long branch chain. Okay, we will have long and unbranched change. While amylopectin, you will have a shorter chain. Okay, you will have a shorter chain than amylose. But they branch up. Okay, you have a long amylose unbranched chain, while amylopectin, you have a shorter chain than amylose, but branch out. Okay, and number 3, they are actually okay. This one extra lah. They are actually curved and coil into helix structure. Helix structure means twisted. Helix structure means twisted. Okay, like the tornado. While this one you have your branches. Okay, this one your branches, they are formed by 1,6 linkages. So this is your amylose, your amylopectin summary.
Okay.
Okay, and then after that, okay, we look at glycogen. So glycogen also easy lah actually. Glycogen, they are also called storage polysaccharides, but they mainly in animals and fungi. Okay, they are highly branched, but they does not coil. Okay, they won't coil. And you need to know a very important thing. Glycogen is stored in liver and muscle cells. Okay, liver in muscle cell. That's why we also got glycogen. Why? Because glycogen is like during, okay, if you are low blood glucose level during that time, maybe sometime, okay, example, Muslim will have fasting, right? Okay, I guess you have fasting period, I think a few months before. Yeah, so if you don't eat the whole day lah, okay, we won't, okay, to be straightforward lah, we won't die, okay? I use this word, we won't die, and we won't like cannot survive. Why? Because your liver will have glycogen. So the glycogen is like a polysaccharide. So they will digest, they will detect the level of your blood glucose and do some work. Okay, so they will release some of the glucose molecules to your bloodstream and travels to your cells or tissues so that you can get energy because they undergo respiration. That's why glycogen, they are stored in liver and muscle cells. Okay. And then your glycogen, they are actually more branched than your amylopectin and making them more compact, which helps your animals store more. Why? Because animals sometimes during winter season, they will sleep, right? Okay. They got a period like they want to sleep. So that during that time, they will eat and store the food. Or sometimes you heard before a snake, they ate a whole chicken and they can store the energy for 2 to 3 days without eating because they can store their food inside the glycogen. Okay, so this is your glycogen part. And their structure, you see, they got a lot of branch. If you compare to amylopectin. So you see, they can branch all things. Okay, so this is the feature lah, the summary feature of your starch and glycogen. So you see, both starch and glycogen, they are formed by alpha glucose. This one important. So amylose is not branch, amylopectin is branch, glycogen is branch. And they will form something called helix shape. Okay, let me find a picture for you for helix shape. Wait, helix shape is like the spring lah. Okay, the spring you are using, you are encountering that one. Okay, wait, paste it here lah. So during revision you can refer. Yeah, this is called coil, or we call it helix. So you see they are twisted like a spring.
Okay, yeah.
Yeah. And your glycosidic bond present is in what? Okay, only amylose is 1,4 lah, because they appears as a straight chain. But starch, amylopectin, and glycogen, you got 1,4,1,6, 1,4,1,6. Because they are chain, okay, because they are chain plus your branch. That's why they are very compact. Okay. Yeah, so this is the table you need to know. Okay. So if you finish finish studying the notes lah, so the table form here will be the best study tools lah, I would say, to help you to revise. So this is the part for starch and glycogen.
Okay.
Okay, then after that, okay, let us look at cellulose. Okay, a new thing here, which is the last polysaccharide lah we will be entering. So cellulose, okay, they are actually called unbranched polysaccharide. Okay, they are actually called unbranched polysaccharide. So unbranched polysaccharide means, okay, they don't have any branch, they're also straight chain, but they are actually formed by, okay, you can jump to here, long chain beta glucose joined together by 1,4 glycosidic bond. Okay, 1,4 glycosidic bond, and a special thing here. Okay, a special thing here. Okay, your consecutive beta glucose molecules, they must be rotated 180 to each other. Okay, so what does it mean now? Okay, let me show the picture below. So first one. Okay, so first one, yeah, you see beta glucose. Okay, beta glucose, here. Okay, this is the normal upright glucose, means that not inverted. Normal upright beta glucose here. So when you want to form glycosidic bond with your next glucose, your OH here, okay, initially the OH should be like that, right? H, OH, okay? But instead, you invert it. Okay, so this one you need some imagination. Okay, let me throw back to the beta glucose we learned before. Yeah, so you see, beta glucose initially is like that, right? OH, H, and this is HOH. So right now you imagine they turn 180 degree. Okay, they turn the whole thing, and they become this one. This one. So is it okay? This one. Okay, imagine. They rotate
Yeah.
180. Yeah, the
Yeah.
mathematics, you learn the plane, right? Yeah, the plane is like they turn the whole thing. So example is like that. Example, I put one thing lah, okay? Maybe I put one sticker. Yeah, I think this one, I put this one. So you see, if I rotate, if I rotate 180, right? Yeah, so this is the inverted. Just now is this one, right? Then I go back to this one. This is 180, and this one I turn 180, turn like that. Yeah. So beta cellulose is special here. Okay, cellulose is special here. So this
Okay.
one is our part here.
Yeah.
And after that, to form the glycosidic bond, you need a lot of hydrogen bonds here lah. Okay, to give their strength. That's why cellulose, if you know, cellulose, they will make up the cell wall of plant. This one, do you know?
Yeah, I do.
Yeah, okay. Got impression, right? Yeah. So they make up of cell wall of the plant. And we learn in IGCSE, cell wall, their structure is strong and rigid. Okay, strong, or you want to read it, pronounce it as rigid or rigid also. Yeah, that's why they help the plant to withstand cell wall. That's why they are strong, because they got a lot of hydrogen bonds. So this is beta glucose for cellulose. So what you expect is, you see, I go down, I go up, down, up, up, down, up, down. Okay, I go up and I go down, up and down, because inverted嘛. So this causing them to be very strong. Okay, you see, I got two layers. One is the first layer, another one is like the second layer. So you see, you can imagine as your phone lah. Normally we go for one screen protector, right? Cellulose is like they got two screen protectors. So this caused them to like stronger. Okay, stronger. Okay, so this one is our example lah.
Okay.
Yeah, so if okay, cellulose, okay, the function you see, main structural component of your cell walls, and they will have high tensile strength. Tensile strength means that the elasticity lah, you can lengthen or shorten. We call it tensile strength. Okay, and they will have a strengthened cell wall to support the plant. And their cellulose fiber is freely permeable. Okay, take note here, cell wall, we call it fully permeable. Means that I let everything come in or go out from my cell. This is what we call cell wall. And we got another one is cell membrane. Yeah, cell membrane, we call it partially permeable membrane. So fully permeable membrane means that I let something come in and come out. Everything, almost everything. But cell membrane is like I only allow certain substances to come in or go out. So this is the cellulose function. Okay, cellulose function. So this is their diagram lah. Okay, you can read through here. Okay, so we are done with cellulose. Okay, we are done with cellulose.
Okay.
Yeah. Yeah, so if here okay. Okay, we look at lipid. Okay, the last part lipid, then we take a second break. Yeah, so lipid, let me see. Can I do it? Oh, still got a lot. Okay. lipid here. Okay, I enter the first part here. Okay, lipid. Remember, carbohydrate. Okay, carbohydrate and lipid. Okay, carbohydrate and lipid. Okay, you need. Okay, both of them are made up of carbon, hydrogen, oxygen. They are the same thing. Okay, lipid, they are actually carbon, hydrogen, and oxygen. So they are CHO. And they are actually hydrophobic and insoluble in water. So what is hydrophobic? Hydro is water. Phobic, we call it phobia. So phobia is you scared water. Okay, you scared water. That's why your is insoluble, right? Okay, that's why your oil, they will float on the water. Okay, yeah, so they are hydrophobic and they consist of CHO. So examples is like fats, oils. Phospholipids is they made up of cell membrane. Okay, they made up of cell membrane. And we got steroids and wax. So steroid is you can found in some of the medication. Okay, but too much steroid is not so good lah. Okay, too much steroid is not so good. Okay, and then we will have, okay, the first one we will have triglyceride. Okay, so triglyceride, okay, they are actually non-polar, hydrophobic. So non-polar, they often come with hydrophobic. So polar, they are actually hydrophilic. So you can take note on this one. They are actually a combination. Okay, they are actually a combination. And the monomers, they are glycerol and fatty acid. Okay, glycerol and fatty acid. And okay, this one you need to know here only. The remaining one you can read through. Okay, the remaining one you can read through. And okay, here this is the diagram they show you. So okay, if you got only single bond, so you see all of them is single bond. Single bond. Okay, single bond. So single bond means that you are saturated. Remember this word, saturated. So you remember S, single bond. Okay, you remember S as your single bond, while double bond is unsaturated. So unsaturated, you see the U here, I macam got equal part, right? So this is what? Double bond. Or sometimes triple bond also called unsaturated. Okay. And both of them got methyl group lah, and also carboxyl group. Okay. Yeah, and this one more than one double bond, we call it polyunsaturated. Means that they got more than one double bond. So this is called monounsaturated, polyunsaturated, and this upper part is called saturated. Okay, so
Okay.
this one is it okay?
Yeah.
This one okay, huh? Okay.
Yeah.
So if this part okay, okay, if this part okay, I think uh I think we can take a short break lah. We can take a short one, 3 minutes break. Okay. Then we continue with triglycerides. Because today a bit packed lah, the knowledge. Okay, then let us continue the remaining part. Okay, so actually, same thing. Triglyceride, how do we form? So you will take one glycerol, three fatty acids. You combine them together by which method? Same thing, we call it condensation. So how do we do? I throw the H here, I throw the OH here, and I form three water molecules. Okay, I form three water molecules, and I will get my triglycerides. So this is the concept lah, I think. Okay, this is the concept. So every condensation, just you throw one H and OH, then you can get your answer already. If they ask you to draw the structure lah, normally they will give you a structure, no need to memorize. They just want you to combine. Okay, they just want you to combine. Yeah, so you see, glycerol and three fatty acids, you condensation, you form triglyceride. And between them, we call it ester bond. Okay, we call it ester bond. Why? Okay, chemistry. Glycerol is actually a type of alcohol. Fatty acid is a type of carboxylic acid. So when these two things combine together, you form something called ester. Okay, you form something called ester. So this is the triglycerides part. Yeah, and the function very easy. Okay, you act as energy storage. Okay, you act as energy storage. So you will have something called long hydrocarbon chain. Okay, why? Because when the bonds between the hydrocarbon chains are broken, they will release energy. Okay, they will release energy. And for your information, triglycerides, they will have more energy per gram than your carbohydrate and protein. Okay, or in the simple words, lipid will have more calories than your carbohydrates and proteins. That's why you will say you will encounter like they say don't eat so much oily food. Yeah. Why? Because okay, in terms of nutrients lah, in the pharmacy we learn lah. Okay, 1 gram of protein, 1 gram of carbohydrate. Okay, they will give you what? They will give you 4 kilojoule of calories. But you imagine 1 gram of lipid, you will get 9 kilojoule of calories. So you see the difference. I eat 1 gram of lipid, I get twice more than the calorie of your protein and carbohydrate. That's why eat oily foods will get fatter easier. Okay, so this is the energy storage. Okay. So triglycerides, they are actually hydrophobic, right? So they will cause osmotic water uptake. This one is not so important. You can just remember energy storage. And they will act as insulator lah. Okay, they will act as heat insulator. Or we call it insulator. Can already lah. Why? Because during cold weather, those with fatty adipose tissue lah, they won't feel so cold. Okay, why? Because they got enough energy to give them the temperature. Okay, but this is not the reason for you to eat so much. Okay, so this is the concept here. Okay, and a special thing is what? Okay, oxidation of your carbon hydrogen bonds, they will release large numbers of water. Okay, they will release large number of water. This one we call it metabolic water. Okay, we call it metabolic water. So example like some animals in the desert. Okay, they will retain this water because they don't have liquid water to drink. So normally if you observe those desert animals, they are actually large in size, like camel, those things, right? They are actually, their size is larger, a lot of fats. Why? Because if they metabolize their fat, they can get water. Okay, they can get water. And second thing we have is bird and reptiles. So bird and reptiles in their shells, they also use this water. Okay, they also use this water. Okay, so this one is the part here. And some of the plants, they store triglycerides in the form of oil in their seeds or fruits. For example, you will saw olive oil, sunflower oil, and other things. Okay, so this is our energy storage. Okay, this is our energy storage.
Okay.
Okay. Oh, here we got act as insulation. So they are actually part of our composition of myelin sheath. Okay, myelin sheath, you will learn it in neuron chapter. Okay, so they will provide insulation, which increases the speed of transmission of nerve impulse. Yeah, so myelin sheath, their function is to increase the speed of transmission of your nerve impulse. Okay, they will act as our insulation. And triglycerides, they will store in adipose tissue. So adipose tissue below the skin, which act as insulation against the heat loss. Okay, so what is adipose tissue? Okay, you try to use your finger lah, to pinch your arm. Okay, you pinch your arm. So the part that you pinch is what we call adipose tissue. Okay, so they act as insulation lah, okay, to help you to retain during cold weather. Like example, rainy days, you won't feel so. Okay, they will help you to insulate lah. Okay.
Okay.
Okay. Then after that, we have buoyancy and protection. So low density of fat tissues, they will increase the ability of animals to float more easily. Okay, this is something about physics already. And protection, okay, very important. Actually, fats help us to protect goods from the risk of damage. But don't take me wrong. Too much oil surrounding the organ is not so good. Yeah, but you need to have at least some of it in order to protect your organ. So this part, is it okay?
Yeah, it's okay.
Okay, yeah. And after that, the last part, okay, before we end our class. So okay, the important role of your phospholipid. So phospholipid, they are actually also formed by your lipid lah, okay? And they also got glycerol and fatty acid. So you got something called phosphate group. Okay, you got something called phosphate group, which is the blue color part. So this phosphate group, they are actually polar, while the fatty acid tail is non-polar. Okay, phosphate is polar, fatty acid tails are non-polar. Okay, so you will learn a word called amphipathic. So means that phospholipid, they both have hydrophobic and hydrophilic part in the structure. We call it amphipathic. This one need to remember. Okay, this one need to remember. So as a result of having hydrophilic and hydrophobic parts, they will have monolayer or bilayer in water. That's why normally we say phospholipid bilayer. So imagine this is the head, this is the tail. Okay, okay. Then you will have a tail here and the head. This one you'll learn in the new chapter. So you see, I keep on multiply, you will see the difference here. The head is towards outside, while the tail is inside. So this is what we call phospholipid bilayer structure.
Okay.
Okay. Then after that, okay. So you see, we got mono layer and bilayer. So normally we will have mono layer lah. We will have bilayer. So this is their head heading outside. One is heading inside. So normally the middle part will be our hydrophobic tail. Okay. And lastly, the role of your phospholipid bilayer. Okay, first thing, they contribute to fluidity of the cell membrane. Okay, fluidity means that how smooth. Okay, how smooth the membrane. So if they are actually saturated fatty acid, you will be less fluid. Means that less flexible lah. While if you consist of many unsaturated fatty acid, you will be more fluid. Okay, so this is the main part of your role of phospholipid bilayer. Okay, so phospholipid second function, they will also control the membrane protein orientation. So weak hydrophobic interactions between phospholipids and membrane protein, they will hold the proteins within the membrane, but still allow the movement within the layer. Okay, so this is the role of phospholipids. Okay, the role of our phospholipid. And then, okay, the table, this one, just need to roughly know lah, difference between phospholipid and triglycerides. So phospholipid, I got 2 fatty acid tail. Triglyceride is 3, right? So I got 3 number of fatty acid tails. And phospholipid got phosphate because phosphor, but triglyceride don't have. So if you got 2 fatty acid tail, you can form 2 ester bond. If you got 3 triglycerides, 3 fatty acid tail, you can form 3 ester bond. Okay. Polar and non-polar. Okay, polar, you got polar phosphate head. This one is non-polar. And number of water molecules, this one special. Both of them got 3 water molecules. And function of your cell membrane component, your phospholipid, they involve in cell membrane, while triglyceride help you to store your energy. Okay, yeah, so we are done with, today we did a lot lah. We got carbohydrate and protein. Okay, so this part is okay?
Yeah.
Okay, yeah, so if this part okay,
Yes.
later I'll send you the recording.
Okay. Thank you, teacher.
If you don't have any question, then we will end here.
Yeah, I don't. Okay. Thank you, teacher.